Hello, everyone. Today, I would like to share with you our recent research titled Nano-plastics Disrupt Systemic Metabolism by Remodeling the Bile Acid Microbiota Axis and Driving Hepatic Intestinal Dysfunction. This study investigates the mechanisms by which chronic ingestion of environmentally relevant doses of nanoplastics impacts host systemic metabolism and the gut-liver axis. Microplastic pollution is a pervasive health risk with particles detected even in human blood and meconium. While single-organ toxicity like intestinal inflammation is known, we lack a clear understanding of how microplastics disrupt the gut-liver-kidney axis to cause systemic metabolic collapse. This study aims to bridge this gap, focusing on the mediating roles of bile acids in the gut microbiota. For the experimental design, we established a chronic exposure model. Male IC or mice were exposed to 200 nanometers PET nanoplastics via drinking water at 15 microgram ml for 9 weeks, mimicking environmental low-dose exposure. We then employed a systems biology approach combining histopathology, biochemistry, targeted metabolomics, and metagenomics. The results reveal a distinct systemic phenotype. The mice exhibited hyperarousal but progressive weight loss and organ atrophy. Biochemically, we observed significant hyperglycemia and dyslipidemia, with elevated triglycerides and cholesterol. Metabolomics pointed to constrained mitochondrial oxidative phosphorylation, forcing a shift to glycolysis, leading to a maladaptive hypermetabolic state driving systemic collapse. At the organ level, we observed severe lipid accumulation and inflammatory infiltration in the liver and kidneys. Q or PC or data suggests that activation of an FKB and an L or P3 pathways suppressed fatty acid oxidation while promoting lipogenesis. Simultaneously, the intestinal barrier was compromised with mucus depletion and elevated oxidative stress. This inflammation metabolism crosstalk is central to the observed organ pathology. A core finding is the profound remodeling of the bile acid pool. Nanoplastics hyperactivated the hepatic CYP7A1 pathway while blocking microbial 7-alpha dehydroxylation. This caused an accumulation of cytotoxic primary conjugated bile acids, like TCA, and a depletion of protective secondary bile acids, like LCA. This imbalance directly fueled hepatic lipogenesis and exacerbated intestinal inflammation. Chronic exposure significantly reduced bacterial alpha diversity, while conversely increasing fungal and viral diversity. Neutral model analysis revealed that bacterial assembly was driven by strong deterministic processes with reduced dispersal. Interestingly, fungi and viruses exhibited higher stochasticity, suggesting their growth was an opportunistic expansion following the collapse of the bacterial community.
We observed a significant depletion in clades associated with SCFA production and 7-alpha dehydroxylation, such as lineages containing Ruminococcus and Lachnoclostridium. In contrast, the enriched taxa, like Prevotella and Bacteroids, mapped primarily to carbohydrate fermenting and mucin-degrading evolutionary clades.
Notably, fungi like Aspergillus and retroviruses expanded significantly. Beyond community structure, we also observed profound remodeling at the functional gene level, specifically regarding carbon metabolism enzymes and antibiotic resistance genes. First, the carbohydrate-active enzymes, KZ, profile shifted towards a destructive state. We observed a significant increase in glycoside hydrolases, GHs, particularly those degrading host mucin glycans, like GH1 and GH81, while glycosyltransferases, GTs, which are crucial for maintaining the mucus barrier, significantly decreased. This indicates a functional shift from barrier construction to mucin degradation, effectively eroding the host's physical defense. Second, a surprising finding was the comprehensive contraction of the gut rhizostome. This suggests that chronic nanoplastics exposure renders the gut ecosystem extremely fragile, compromising both the physical barrier and the ability to withstand external stress. In conclusion, chronic ingestion of nanoplastics does not just damage the gut but triggers systemic metabolic collapse and multi-organ dysfunction by remodeling the bile acid microbiota axis. This study provides a novel mechanistic framework for understanding NP toxicity. Moving forward, we need to explore therapeutic interventions, such as modulating bile acid metabolism or using probiotics to mitigate these effects. Thank you.